Hello everybody, here we go. This is the final review for the class. I'm going to do a little bit of review of the nervous system in advance of your last exam, last physiology exam for the course. I'm not really going to cover much of the anatomy in here. That just takes lots of practice. Be sure you use the Unit 5 Nervous System Anatomy Practice Test that's posted for you. That is a big time hint. Be sure you do that multiple times before you take the actual Unit 5 Nervous System Anatomy exam. Should be a good chance for you to make a high score here at the end of the semester. Nervous system physiology is a little more complex. I think the most difficult part of the nervous system was learning about how action potentials were generated, and we've covered that in Unit 4 already. But no doubt the nervous system is pretty complex, and you guys no doubt have probably noticed in your textbook we didn't even cover a lot of the information in those chapters, so it's even more complex than what we've gone over. Just lots of brain work to get through the nervous system. Nervous system obviously though is really important. Okay, nervous system organization. So you have an anatomical way of dividing up the nervous system, and that's where you divide it into the CNS versus the PNS, central nervous system and peripheral nervous system, central nervous system, brain and spinal cord, peripheral nervous system, cranial and spinal nerves. And then you have a functional division as well. So the sensory division of the nervous system, that involves neurons that are generating action potentials headed towards the central nervous system. So that's incoming information toward the brain and spinal cord. The motor division is outgoing information, so action potentials that are starting in the central nervous system, passing through the peripheral nervous system out to control skeletal muscles, glands, major organs. That's your motor division. Now your motor division has two branches. It can be divided up into the somatic nervous system. That's what we studied in Unit 4, the motor neurons controlling your skeletal muscles. And that's voluntary, under voluntary conscious control in many or most cases. Autonomic nervous system, auto means like automatic. So you don't have to think about what this branch of your nervous system is doing. You don't have conscious control over this. So this involves control of glands and organs and things like body temperature as well. This has two divisions, so you just got to kind of outline all of this. You've got a sympathetic division and a parasympathetic division. The sympathetic division is sometimes called fight or flight, so it's involved in stress or survival responses. And your parasympathetic division is sometimes called rest and digest, so it's returning you to a normal state. So again, there's just some memory work there. All right, practice with the terminology. Which branch of the nervous system transmits nerve impulses to your skeletal muscles? Okay, and remember we got somatic refers to the body. A lot of times here, like in this case, it's referring to those skeletal muscles. Visceral refers to internal major organs, your viscera, like your stomach and your small intestine and your liver and your urinary bladder, things like that, your viscera. So which branch? Hopefully you're thinking somatic motor, because motor is outgoing impulses toward those skeletal muscles. That's going to be your somatic motor nervous system. Which nervous system division increases activity during times of stress? So we just talked about that. It's a branch of your autonomic nervous system, involuntary, and this branch increases during times of stress to help you get through stress or survival type situations. Hopefully you're thinking the sympathetic division. I know the terminology can be kind of weird. Sympathetic, parasympathetic, what in the world does that mean? But so you just have to kind of memorize it. And I don't want to imply that the sympathetic division only gets activated during times of extreme stress, just anxiety. Anytime you need to increase your blood pressure or your breathing rate to help you maintain homeostasis, the sympathetic division is going to be involved. Anytime you need to lower your heart rate lower your breathing rate, return to a normal state, help you maintain homeostasis for things like blood pressure, breathing rate, heart rate. The parasympathetic division gets involved in that. But to help you learn it now and remember it for later, sympathetic, fight or flight, parasympathetic, rest and digest. And you guys know what goes on with when you are anxious or frightened, your heart rate goes up, your breathing rate goes up. So that can help you keep those things straight. All right, nervous tissue, you do want to go back and think about the different components of nervous tissue that we learned about. And of course, the neurons are the main functional cells of the nervous system. Those are the ones that generate the action potentials or nerve impulses. And remember, they try to control each other. Then you have neurons that control skeletal muscles and glands and blood vessels and internal organs and so forth. But then you also have other supporting cells that are part of your nervous system as well. And on our diagram here, we see the ependymal cells. Those are the cells that line the ventricles in the brain. And so they're going to be facing, one side will be facing cerebrospinal fluid in the ventricles and the aqueduct and so forth. And the other side is just facing the interstitial fluids within your central nervous system tissues. This is a diagram of central nervous system tissue. Of course, you got capillaries that bring the blood to your nervous tissues are very metabolically active. They got to have a good blood supply, so you're going to have blood vessels in there. Astrocytes are these star-shaped cells that a lot of times you find those connecting neurons to the capillaries, and they kind of help regulate what gets passed out of the capillaries and onto the neurons, among other things as well that they do. Oligodendrocytes, there's one of those, and they have branches, and they create the myelin. They produce the myelin insulation around the axons of your neurons in the central nervous system. Remember, in the peripheral nervous system, you have those Schwann cells that do it, and the cells actually themselves are wrapped around the axons. But in the central nervous system, these oligodendrocytes, the cell body is not attached to the axon itself. It sits out by itself. And a single oligodendrocyte can actually send out little projections here that wrap myelin around the axons of more than one neuron, as you can kind of see there in the picture. Microglia, those are cells that help fight infections in the brain. They help clean up debris. They're kind of like macrophages that we learned about earlier in the semester, kind of like vacuum cleaners to help get rid of things like if you have bacteria or viruses. Hopefully you don't, but they help fight those types of things. And they'll also help clean up damaged tissues and so forth in the central nervous system as well. All right, and remember we have structurally or anatomically, there are different types of neurons based on their structure. And what you mainly learn about, most neurons look like this. They're called multipolar neurons. And the poles are referring to the big extensions that extend away from the cell body right in here. So a multipolar neuron has lots of dendrites and then one long axon. The axon may branch like you see there, but there's just one long axon. It's called a multipolar, if you think about each extension being like a pole. Unipolar neuron, this is what most sensory neurons look like. The cell body, it looks like an octopus here in this diagram. It's sitting off to the side, and then there's one long axon. So they're showing you the direction of the action potential, how it travels along the axon. So it generally has little branches over here on one end. These are sensory nerve endings for various things, like this might be a neuron that senses pain, for example. Tissue damage to cause you to feel pain. That's going to be a unipolar type neuron. Remember, you've also got sensory neurons that sense things like temperature, and those have this type of structure as well. And the action potential gets generated because of some stimulus down here, travels towards the central nervous system. Usually these are going to head toward the spinal cord. And then bipolar neurons, these are the ones that are the least frequent. The cell body is sitting in the center. Soma is another name for body. And they've got one dendrite. Sometimes it's referred to as a second axon over here. And then there's a second one over here that may branch. So bipolar, two main extensions away from the cell body. All right, which of the following is true? Go back and review the characteristics of sensory neurons. Select all that apply. So let's just go through these. Remember, with a multiple answer question, you want to just go down the list and treat it like a true-false. The somas are located in the cerebrum. So where is the cerebrum? Hopefully you're remembering that that is the main part of the brain. No, we did not learn that the somas or cell bodies are located there. Most of them are located in the dorsal root ganglia off of the spinal cord. Remember that. Do they have one long axon? Yes. We just talked about that. Do they generate afferent impulses? Don't forget this terminology, afferent versus impulse. Afferent is going towards the central nervous system. Efferent would be away from. So afferent toward, efferent away from. So yes, their nerve impulses, action potentials are headed toward the central nervous system. Do they generally synapse with interneurons in the posterior horn of the spinal cord? Yes, if you go back and look at what we've talked about, the synaptic knobs on the ends of the axon of a sensory neuron are going to form synapses generally with interneurons that are located there in the posterior horn of the spinal cord. So you got all this networking between neurons. Ultimately, it's going to send information up to the brain in most cases, so the brain can make decisions about responses that need to be made. The perikaryones are located in the dorsal root ganglia. Perikaryon is another name. I don't generally use it very much, but it's one of those terms you should know. The perikaryon is another name for the cell body, and we just said those are typically located in the dorsal root ganglia. Are they generally bipolar? No, some of them are, like the neurons in the retina of your eye, for example, but most of them are unipolar. So B, C, D, and E. All right, how about this one? Which of the following is false? Astrocytes bridge neurons and capillaries. So we just took a look at that, and yes, they do that, and probably help transmit nutrients and wastes from one to the other. The oligodendrocytes produce myelin sheathing in the CNS. Yeah, we just talked about that. Microglial cells ingest and digest microbes in the CNS. Yeah, we just talked about that. Microbe is another term for microorganism, like bacteria, viruses. Ependymal cells line the sulci and fissures in the cerebrum. Now that one you have to be kind of careful with. No, remember we said the ependymal cells are special cells that line the ventricles where you have the cerebrospinal fluid. So your sulci and your fissures are just those hills and valleys, those wrinkles and so forth on the surface of the cerebrum. So that's false. Astrocytes reuptake some neurotransmitters. If you go back and look at some of the functions of astrocytes, that's one of the things that they do. They reuptake some of the neurotransmitters. Remember, that's important. Remember, you have to control your neurotransmitter levels so that you don't overstimulate neurons or so reuptake is one of the mechanisms for controlling those levels, and astrocytes can be involved in that. All right, neurons, excitation and inhibition. So we've talked about, here's a neuron. This is the cell body, or the soma, or the perikaryon. Unfortunately, it's got multiple names. And remember, here's your axon hillock, and here's the first part of the axon. The rest of it's cut off. And so what is going to cause this neuron to generate its own action potential, that all or none wave and change of voltage that goes from one end to the other? What is that? What controls that decision-making process? And so we talked about how An individual neuron may be receiving information from multiple other neurons. And that's what they're showing you here. Like, this is supposed to be the synaptic knob of one neuron. Here's another one. Here's another one, and here's another one. And all four of those are trying to influence this one, trying to control whether or not it's going to reach that threshold, that minus 55 millivolts in most cases, and generate its own action potential, like we learned about in Unit 4. So these would be called presynaptic neurons. Here's your synapse between the two. Presynaptic, this is called a postsynaptic neuron. And remember that the neurotransmitters that are getting secreted from the ends of these axons, those synaptic knobs there, or axon terminals, are either going to be inhibitory or excitatory. Excitatory means you're depolarizing locally. Inhibitory means you're hyperpolarizing. So that's why they're showing you here on the graph, a little hump upward, that's depolarization locally. Inhibitory, you're hyperpolarizing. Your voltage is dropping down even lower than normal. And it's the sum of all of those together. If you have lots of excitatory stimulation, eventually that stimulation is going to make its way over here. You're going to, the voltage is going to go up enough here at the axon hillock to trigger an action potential. If you have too much inhibitory stimulation going on, inhibitory makes it less likely that you're going to reach threshold over here at the axon hillock. So it's the combination of all that influence that determines that combination of excitatory postsynaptic potentials and inhibitory postsynaptic potentials that ultimately determines whether this neuron is going to generate its own. So all this is going on all the time, and all this networking that exists in the nervous system between neurons. All right. And remember, an EPSP, excitatory postsynaptic potential, is just a change in voltage in a little local area. So you have to have lots of those adding up before the neuron will be stimulated enough to generate its own action potential. So an EPSP definitely causes an action potential on the postsynaptic neuron. Hopefully you're thinking no there. You got to have multiple EPSPs that have to add up before a neuron is going to generate its own action potential. Causes local depolarization of the postsynaptic neuron membrane. Yes, that's true. In the little local area, voltage is going up. Does it hyperpolarize locally the postsynaptic neuron membrane? No. Hyperpolarization means the voltage is going down, and that's the opposite of depolarization. And that's not excitatory, that's inhibitory. Makes an action potential on the postsynaptic membrane less likely. No, excitatory is going to make an action potential more likely. A neuron cell membrane becomes locally hyperpolarized near a synapse. What has occurred? All right, if you read through those four possible answers, hopefully you're thinking this one, an inhibitory postsynaptic potential. Now you might have to think about C here, an influx of lots of sodium ions, but remember, when sodium comes inside, that makes the inside of the cell less negative because sodium is positively charged, so there your voltage is going up. And that would be depolarization rather than hyper. Hyperpolarization means you're becoming even more polarized. The inside is becoming even more negative than it originally was. All right, which neuron will generate faster action potentials? Which are faster? If you go back and review, not all axons of neurons have been created the same. One with myelinated thick axons, myelinated thin, unmyelinated thick, unmyelinated thin. All right, so now hopefully the first part is easy, myelinated versus unmyelinated, because we've talked about how myelin helps insulate the axon and causes you to only have to regenerate the action potential at the gaps. Remember the nodes of Wollenbier? So action potentials travel much faster on a myelinated axon. It helps prevent the loss of the voltage change. It works like insulation, just like insulating a wire. It helps you maintain the voltage in a wire. And you just have to remember thick or thin, and hopefully you're thinking thick. Action potentials actually travel faster down the thick axon than a thin one. I don't think the thick versus thin is quite as obvious. You just have to remember that one. The neurotransmitter story, we heard about this in Unit 4 a lot, so I'm not going to rehash this too much. But notice, in Unit 4 we were talking about a motor neuron forming a synapse with a skeletal muscle cell or fiber. And in Unit 5 we've been concerned more with neurons networking with other neurons. And just keep in mind the principles are essentially the same. An action potential travels down the axon of your presynaptic cell, as you see here in the diagram. And as that action potential reaches the axon terminal here, a voltage-gated calcium channel opens. That influx of calcium ions causes these vesicles that are storing neurotransmitters to travel up here to the surface. They spit out their neurotransmitters. Those attach to neurotransmitter receptors that are on your postsynaptic cell membrane over here. Those are generally, not always, but generally ligand-gated ion channel. Ligand, the ligand or ligand is just referring to the neurotransmitter. When that binds, it causes the channel to open. And then that allows sodium to flow into the cell and causes depolarization. So right in here, if this was a channel that was opening up and letting sodium come in, that would cause local depolarization there, and that would be an excitatory postsynaptic potential that was occurring there locally on that part of the membrane. All right, select each of the following that is a way of limiting the amount of neurotransmitter molecules in a synapse. So keep in mind, just like we learned in Unit 4, you have acetylcholine esterase as an enzyme that's always hanging out here in the synapse, and it quickly digests any acetylcholine that's trying to stimulate a skeletal muscle fiber, so that you just don't keep re-stimulating that skeletal muscle fiber unless you need to. So there are other ways to make sure that these neurotransmitters don't just hang out in here for anything but a brief period of time. And again, that just helps us keep the nervous system under proper control. Reuptake by the presynaptic neuron. This is one of those select all that apply. Yes, we've learned about that before. That was the way. We've talked about that. Presynaptic neurons can reuptake neurotransmitters. Postsynaptic neurons generally don't, so we'll X that out. Diffusion out of the synapse, just that natural diffusion from higher concentration to lower. Absolutely. Degradation by enzymes, yes, just like we learned about in Unit 4 with the acetylcholine esterase enzymes that chop up the acetylcholine and destroy it. Fewer action potentials generated by the presynaptic neuron. Yes, if you fewer action potentials, that's going to trigger less release of neurotransmitters, so we're going to check that one off. Low calcium levels in the tissue fluid surrounding the neurons. I would say yes on that one as well. You got to have the calcium coming out of the tissue fluids and into the synaptic knob to trigger the neurotransmitter to be released, if you go back and look at the diagram. So we'll do A, C, D, E, and F on that one. All right, this neurotransmitter is important for reward pathways and motivation. So go back and review the video lecture, the little bits of information we talked about the different neurotransmitters. Follow your study guide carefully. You don't have to drive yourself completely nuts trying to memorize the functions of all of these different neurotransmitters. We're just talking about specific examples. Go back and do the mouse party, which is kind of fun to do anyway, and that'll help you learn some of these as well. So reward pathways and motivation. Hopefully you're thinking dopamine. Dopamine is, a lot of recreational drugs that people take to get high are involved with making sure you keep more dopamine around. So all these reward pathways and so forth are stimulated so that you feel good for longer periods of time. A little bit of a review on neuron networking. This would be a very simple neuron network. So remember here in blue, this is supposed to represent a unipolar sensory neuron. That's its little cell body over there. And that would be living in the dorsal root ganglion. This is a cross-section through the spinal cord, of course. You got a receptor on this end that detects some sort of stimulus, like that could be touch, it could be pain, whatever it is. Pressure generates an action potential, and then this neuron forms a synapse with this green one. This is called an interneuron. It's like a bridging neuron. So you got a synapse right there. If this interneuron gets stimulated enough, it generates an action potential and secretes its neurotransmitter that will stimulate this motor neuron, and its cell body is living over here in the anterior horn of the spinal cord. And it will generate an action potential outward, and then it's going to function as an effector that's going to trigger some response. So typically what we have studied is that these neurons are controlling skeletal muscle, so it would be stimulating muscle contraction of some type. So that's a very simplistic neuron network. In many cases, these networks are going to head up the spinal cord and toward the brain so that the brain takes in information and can make decisions. All right, these neurons send outgoing impulses as part of the motor and autonomic divisions of the nervous system, which is our best answer there. And hopefully you're picking motor neuron. So not all motor neurons control skeletal muscles. You have motor neurons that are involved in the autonomic nervous system as well, that control glands and organs. These neurons receive impulses from other neurons and play roles in integration and association. So these are kind of your bridging neurons. Hopefully you're thinking interneurons on those. Okay, the brain. Go back. This is another case where you want to use your study guide for the Unit 5 Physiology exam very carefully. Go back, and your textbook goes into lots of detail on the different parts of the brain, and you want to just kind of cut it down to the basic information that we went over in the video lecture. So use your PowerPoint notes and use the study guide to help you review. Unfortunately, you do have just some memory work here to learn some of the functions of the different parts of the brain and what they do. And then some example questions. This part of the brain plays a major role in developing blueprints for smooth, coordinated muscle movements. And hopefully you're answering cerebellum. That's what the little mini brain on the underside, back, posterior portion of the brain. What that lobe does is, that's one of the things that it does. It develops blueprints for smooth, coordinated muscle movements. So your cerebellum, when you first learned how to roller skate, it was very awkward at first, but once you learn, you tend to remember it. I mean, if you pick it up again when you're my age, it takes you a little while when you first put those skates back on, but then pretty quickly you can start skating. Maybe not as well as when you were 12 years old, but you can pick it up again fairly quickly. And a lot of that is because of the cerebellum has created those blueprints and remembered those blueprints for those smooth, coordinated muscle movements that you need to make that happen. This part of the brain helps you recognize or recall patterns, faces, colors, etc. All right, so hopefully we can tell that we're talking about sight here. So that's going to be visual. And whenever you're referring to a part of the brain that's involved in recognition or calling up memories about some sort of sensory info, that's going to be an association area. So our answer here is going to be the visual association area. The primary visual cortex is going to be the part of the brain that's involved with taking in that information that's traveling down the optic nerves from the retinas of our eyes and creating those images in our mind. That's the primary visual cortex. The association area is what's going to help you realize, oh, I'm looking at John, or I'm looking at Jane, or I'm looking at a tree, or I'm looking at my dog Fluffy, or whatever, helping you recall those types of things. Most of the axons in ascending spinal cord tracts, now again, your textbook has a lot of information on these ascending and descending tracts of the spinal cord, and use your study guide and the information we went over in the lecture to kind of guide you on that. If you know your anatomy, you can figure out what a lot of these tracts do and where they go just based on how they're named. Most of the axons in the ascending spinal cord tract synapse with neurons in this routing station. So you got axons that are traveling up through the spinal cord, they enter the brain, and then you got a part of the brain that receives those impulses, and then the neurons there make decisions about where up here in your cortex information needs to go so that the brain can make proper decisions. And so hopefully you're thinking routing station, thalamus. The thalamus is the part of the brain involved with a lot of that. The thalamus is going to be involved like at night when you're sleeping, and you've got all this sensory information bombarding you all night while you're asleep, little sounds and something might be touching you or whatever, and you might, a breeze due to the air conditioner kicking on, and all that sensory information, if you woke up over every little thing, you would never be able to get to sleep. So the thalamus, one of the things it does is helps decide when something is urgent enough that it needs to wake you up. Or just think about like sitting here right now doing this video lecture. Yes, my sensory neurons are detecting that my clothes are contacting my skin. There's a sense of touch there, but the thalamus is probably playing a role in deciding whether that information needs to make it up to my sensory cortex in the cerebrum to make me aware of it. Imagine if you were aware of every place on your body all day long where your clothes were contacting you. You would be driven crazy by that type of information. So the thalamus is a structure in the brain that helps make those types of decisions about where that incoming information is going to go and whether it needs to go there in the first place. Okay, a patient has suffered a stroke in the left hemisphere that killed neurons in the primary motor cortex. What effect do you expect her to experience? So the motor cortex, those are going to be the neurons up here in a little strip across the top of the brain that send impulses downward toward the motor neurons that control skeletal muscles. And if you guys remember, the axons of those neurons cross over from one side of the brain to the other. So the left side of the brain controls muscles on the right-hand side of the body and vice versa. All right, so let's look at the answers though here. This is one of these where you don't want to just pick an answer impulsively. You always want to read through all four choices. What effect do you expect her to experience? A lack of all movements on the right side. That sounds kind of reasonable. Lack of voluntary movements on the left side, lack of voluntary movements on the right side, lack of sensations on the left side. All right, primary motor cortex, neurons that control the motor neurons that send impulses to skeletal muscles. So you might be tempted to pick A, but it's actually going to be C, voluntary movements. You're making decisions up here about wanting to move muscles over on the right-hand side of the body. And so that stroke will have impacted that. You can still have involuntary movement of those muscles because the motor neurons that actually control those muscles are mainly down here in the spinal cord, so they're not affected by this stroke. So reflexes that can trigger those motor neurons to send impulses without input from the brain, those can still happen. But voluntary movement will be affected. You can't think about, I want to move or I need to move these muscles on the right-hand side of the body. That's going to be impacted if your cortex has suffered an injury from a stroke. All right, if an accident victim suffers a severed spinal cord at the T8, T9 level, he can still feel sensation in his legs as long as the primary somatosensory cortex is functional. Do we think that's true or false? T8, T9 level, that's thoracic, so here in the chest. The primary somatosensory cortex up here, those are the neurons that are concerned with receiving this incoming information from different parts of the body and how we perceive it. That's painful, that's hot, I feel that pressure. That's what these neurons up in here along that primary somatosensory cortex are involved in. But if you've had a severed spinal cord, can you still feel sensation in the legs, real sensation? It's going to be false. You're not, because the sensory neurons that would be involved in sending impulses upward in response to pain or pressure, temperature, and so forth, they've been cut off. They have no way. They have no way to communicate with the neurons that are going to send those impulses on up to the primary somatosensory cortex up here. So if you've been severed in the spinal cord, then you're going to lose that sensation. All right, select each of the following to help protect the brain protection here. So here we're going to pull some various things together that we've learned about, both in this unit and even earlier in the semester, about brain protection. Parietal bones, yeah, hopefully you're thinking yes on that. That is why we have those cranial bones in the skull, to help protect the brain. The dura mater, that tough outer covering of the meninges, remember that? Oh yeah. The transverse fissure, can you think of any reason why that's protecting the brain? No, it's just an anatomical feature. The central canal, is that protecting the brain? That's the little passageway down the center of the spinal cord that contains cerebrospinal fluid, so I would say no on that one. Cerebrospinal fluid, yeah, that's one of its jobs, is to help protect the brain. It provides a fluid for your brain to sit in and cushion it to help. You don't want your brain bumping up into your cranial bones really easily. The blood-brain barrier, yes, remember that is the capillaries that are passing through the nervous tissue in the central nervous system are not as leaky. They don't let things pass out of the capillaries and into the surrounding interstitial fluids as easily as in other parts of the body. That's called the blood-brain barrier. Microglia, we've already talked about how those cells help gobble up things that are not supposed to be in our central nervous system tissues. Corpus callosum, is that protecting the brain? That's a crossover point for myelinated axons between the left and the right-hand sides of the brain. All right, so go away. A, B, E, F, and G would be our answers there. Spinal cord reflexes. All right, so reflexes, remember, are things that happen automatically, involuntary, and the brain, the cerebrum, does not have to get involved at all in making those decisions. So you have neuron networks that are involved in taking incoming sensory information, sending it up here to the spinal cord, interacting with interneurons that then form synapses with motor neurons that have outgoing information that gets sent outward, generally for contraction of skeletal muscles. Those are going to be things that are involved in reflexes. Again, keep in mind the routing here, the axons of sensory neurons travel through peripheral nerves that lead to bigger peripheral nerves that ultimately lead to these spinal nerves or cranial nerves, if you're thinking about cranial nerves that contain sensory axons. And then the cell bodies, the axons of your sensory neurons pass through the dorsal root of the spinal nerve and head that way towards the spinal cord. And their cell bodies live in the dorsal root ganglion over here. And the axons of your motor neurons, the outgoing, pass through the anterior or the ventral root and then enter the main spinal nerve. And then they're going to go to wherever they're destined to go in the body from there. And they're showing you here on this diagram too where the cell bodies of these different types of neurons tend to live. So go back and review what we covered on where these different types of neurons exist in the spinal cord, and also what we talked about with reflexes. All right, so a patient suffers an injury that severs the dorsal root of a spinal nerve. What effects do you expect the patient to have? If this got cut off, what's the impact going to be? So you've cut these axons of these sensory neurons, so they can't send impulses anymore up here to the spinal cord. All that sensory information has been lost. All right, A, loss of only somatic sensations in the body region served by the nerve, flaccid paralysis and muscle atrophy in the body region served by the nerve, loss of both somatic and visceral sensations in the body region served by the nerve, loss of all functions, motor and sensory, in the body region served by the nerve. So if we go back and look, the blue neuron, that's a somatic sensory neuron axon, and green, that's a like I just realized they've got the same color on some of these neurons. In darker green here, that's a visceral sensory neuron. And so both types of neurons would be impacted by that. So we would say loss of both somatic and visceral. Visceral sensations are coming from internal major organs. So those would be lost as well. All right, here's a question about a tract in the spinal cord. A tract is an area within the spinal cord in the white matter that contains myelinated axons, and that's why it looks white. And you can have ascending tracts headed toward the brain, descending tracts coming down from the brain. So afferent information going toward the brain, efferent coming downward away from the brain. The corticospinal tract, so go back and review which of these we discussed. And you can tell by the name, cortico refers to the cortex of the brain, spinal cord. So the impulses are starting here and going here. So that information lets you know this is going to be a descending tract, a descending bundle of nerve fibers extending. That's what a tract is, extending from the cerebral cortex to the spinal cord. So our answer there would be A. All right, go back and take a look at what we discussed with reflexes and how sometimes both sides of the body get impacted in different ways. So here we've got this poor guy here is about to step on a tack, and when he does, sensory neurons are going to detect that injury. They're going to be involved in pain sensations as well. They're going to generate action potentials that are afferent, travel along the axon there through the dorsal root, come into the spinal cord. They'll form a synapse with interneurons here in the spinal cord. And the interneurons here are going to do multiple things. All right, so this guy, because he's stepping on the tack, he needs to lift up his leg on that side of the body. So you're going to stimulate the muscles that are involved in flexing the knee, and you're going to relax the muscles, the quad muscles that would be involved in extension. Okay, so does that make sense? If you need to bend your knee, you need to stimulate your knee flexors, you need to extend I'm sorry, you need to inhibit your extensor muscles. Remember, your quads are your extensors, and your hamstrings are involved in flexion. Okay, now over on the opposite side of the body, if you step on a tack like this guy with your right foot, you don't want to simultaneously lift up your left foot because then you'd fall down to the ground. So you need to do the opposite over on this side. So interneuron send impulses over to the other side of the spinal cord to other interneurons, and you're going to have synapses here that are involved in eventually stimulating extensor muscles on this side so that they contract, and you want to relax your flexor so that you don't relax the knee. So that's one of these reflex situations where you have opposite things happening on opposite sides of the body. And it's a reflex. This is an automatic thing. You don't have to think about this. Probably this has happened to your right leg and your left leg before you're even able to sense the pain up here in your cortex. All right, that type of situation is called a crossed extensor reflex arc, where you're doing opposite flexion extension things on opposite sides of the body. Same thing happens with, remember the example of if somebody grabbed your arm over here on the right hand side, without even thinking about it, your reflex is to flex the elbow on this side, and you extend the elbow on this side to try to push your attacker away. That's just a natural reflex type thing you don't even have to think about. All right, which of these is not part of a crossed extensor reflex arc? And if you look through those, only one, the ascending and the descending tracts, those are concerned with sending information back and forth between the cerebrum and the spinal cord. And yes, that's still going to happen, but that's not part of the crossed extensor reflex arc. All that just happens through the spinal cord. Okay, nerve structure. So this review, I think, is getting kind of long. So go back and look at, this is an example of a peripheral nerve. And a peripheral nerve is a big bundle of axons. So there's a single axon, and this one happens to be myelinated. That's what those little blue bubbles are indicating, like we studied back in Unit 4. Within a larger nerve, you're going to have blood vessels providing a blood supply to all these axons. Within a peripheral nerve, you can have incoming and outgoing afferent and efferent axons bundled up together in the same nerves that are traveling to and from parts of the body that are kind of in similar locations. And in yellow here, those are connective tissues. Remember, you've got the endoneurium and the perineurium. And the epineurium, I'm sorry, these connective tissues that surround and insulate these different parts within the nerve. So I'm not going to draw all those on there. Be sure you go back and review the parts of your peripheral nerves. All right, this question: an injury to a dorsal root of a single spinal nerve would impact which of these? A single spinal nerve. Dorsal root, remember the axons of sensory neurons pass through the dorsal root. So hopefully you're thinking sensations from areas served by the nerve. Remember, your axons of motor neurons travel outward through the ventral root. Homeostatic imbalance. This condition occurs when a blood clot in an artery cuts off the blood, that should be cuts off, not cuts of, the blood supply to a region of the brain and kills neurons in that area. Kills the neurons. So is that a transient ischemic attack? Ischemia occurs when you have a cutoff of the oxygen supply to a particular tissue. Okay, so that can be temporary or long-term, but transient means temporary. Is it multiple sclerosis? Is it a cerebrovascular accident? Is it a transection? Transection would be like if you cut the spinal cord, like in an accident. Hopefully you're thinking CVA there, or cerebrovascular accident. That's also another name for a stroke. Transient ischemic attack would be like if the arteries that are supplying the oxygenated blood to an area of the brain constricted and temporarily kind of cut off or limited the oxygen flow to that part of the brain. That part of the brain might temporarily not function very well because the neurons that receive their blood supply from that artery wouldn't be getting the nutrients and the oxygen gas that they need temporarily. And if that continues, they might die. In a stroke, you have neurons that actually die and are no longer functional at all. And that's called a cerebrovascular accident, which is another name for a stroke. All right, some autonomic nervous system questions. Which division of the ANS includes neurons that secrete norepinephrine? Go back and look at the neurotransmitter story for the autonomic nervous system. And so hopefully you're thinking sympathetic. Think epinephrine, norepinephrine, think sympathetic. Another name for epinephrine is adrenaline. And you guys know I'm having an adrenaline rush. So an easy way, that happens when you're excited, when you're frightened, when you're stressed, that adrenaline rush. So just associate that with the sympathetic, the fight or flight branch of the nervous system. The sympathetic trunks that run parallel to the spinal cord, go back and review the information on that. What do they contain? So I'll give you some time to read through the answers there. Cell bodies of ganglionic neurons. So your sympathetic trunk, remember a ganglion is an area outside the central nervous system where you have cell bodies located. So they're within the peripheral nervous system. And so that sympathetic trunk is a little thin chain of ganglia where you have these cell bodies of what are known as the ganglionic neurons. And when they get stimulated, they send impulses, sympathetic nerve impulses, towards your major organs. They control the viscera of the body. Acetylcholine always functions as a stimulatory neurotransmitter. Is that true or false? And hopefully you're thinking false on that one. Acetylcholine, that was an example that we used of a neurotransmitter that can be used in different ways in different locations, and that's because there are different kinds of receptors that respond to it. So remember, acetylcholine is a neurotransmitter. It attaches to its receptor. If that receptor opens up and allows an influx of sodium ions, and that's going to cause depolarization. But neurotransmitters may have different kinds of receptors in different locations that will cause stimulation or inhibition depending on what they're doing at that particular location. So go back and review that information that we covered about acetylcholine as well. Okay, hopefully that was helpful and not too long. If you have questions, please post those on the discussion board, or you can of course message me if it's something private. All right, have a good luck with studying for this final test for the course, and again, let me know if you need any help.